Hi, welcome to this paper run through. Now we're going to be running through the AQA A-Level Biology Paper 1 from June 2018, and the reason we're going to do that is because the 2018 and the 2019 papers probably have content the most similar to papers this year, because if it came up in 2021 or 2022, it's kind of a little bit less likely to come up in 23. Still might do. So that's why we're going to start with this 2018 paper, and then I'm going to put a 2019 paper run through out for you next week. So there's two ways you can do this. You can either use this paper to kind of mark your paper. So I'll post the link to the blank paper, do that blank paper, then come back to this to kind of mark it and just check that your interpretation of the mark scheme is correct, because they are really picky. Second thing, if you're feeling like you're a little bit struggling, you want a bit more guidance, we can kind of go through this paper together. You can either pause, attempt a question, then mark it with me, that sort of thing, or you can kind of just follow me to sort of see how you approach the question. Okay, so let's get into it. Question one. Question one says, Figure 1 shows all the chromosomes present in one human cell during mitosis. The scientists stained and photographed the chromosomes. And then in Figure 2, the scientist has arranged the images of these chromosomes into homologous pairs. So Figure 1 is more without any arranging. So it says, give two pieces of evidence from Figure 1 that the cell was undergoing mitosis. Okay, so we just want to look at this here. So what can we see here? Well, first of all, we can see chromosomes. They are visible. They are condensed, therefore. And that is evidence that we're doing cell division of some type. So that's probably the first thing that I would say, that we can see the chromosomes, they are visible. And the second thing is that when we're looking at this kind of jumble of chromosomes, they are not in homologous pairs. Okay, so these chromosomes here, those are clearly not a pair. They're just kind of randomly arranged, which means that we're probably doing mitosis and not meiosis, because meiosis would be homologous pairs. Let's check out the mark scheme and see if that is what we need to say. Okay, here we are. So first of all, the chromosomes are visible because they have condensed. I think we need to have this whole bit here visible because they have condensed. And second bit, okay, this is something I didn't say. So the chromosome is made up of two chromatids. Okay, each chromosome has two chromatids because the DNA has replicated. And then the chromosomes are not arranged in homologous pairs, which is indicative of mitosis, not meiosis. Remember that when we've got a chromosome like this, one half of it, I'll sort of highlight one half of it is the chromatid, and the whole thing is the chromosome. Okay, you need to understand the difference between chromatid and chromosome if you didn't already know that. All right, let's go back to question 1, part 2. Okay, tick one box that gives the name of a stage of mitosis shown in Figure 1. Okay, so in Figure 1, we've got chromosomes randomly arranged, so we're definitely before metaphase. We're not anaphase, so we have to be in prophase. Interphase is the phase between cell division, so we wouldn't see any chromosomes at all. They'd be being replicated. Anaphase, they'd be pulled apart like this. And telophase, they'd kind of already be, the new nuclei would already be sort of starting to form. 1.3. When preparing the cells for observation, the scientists placed them in a solution that has slightly higher or less negative water potential than the cytoplasm. This did not cause the cells to burst, but moved the chromosomes further apart in order to reduce the overlapping of the chromosomes when observed with an optical microscope. Suggest how this procedure moved the chromosomes apart. So we've got a cell here, we've got a nucleus with the chromosomes. I'll just draw a few like this, okay? So the scientists put it in a solution which has a high water potential. So water moves from a high water potential to a lower water potential via osmosis, but the cell didn't burst. It probably just swelled up in size, and water would enter the nucleus as well. So the cell just swells up in size, which means the chromosomes inside the cell just kind of get pushed further apart. They spread out more. So let's check the mark scheme and see if that is what we need to say. Okay, so you need to say by osmosis. So water moves into the cells. Always say by osmosis. If the question is about osmosis, you need to say by osmosis. Always. If you don't say it, you won't get the mark. So the cell gets bigger, and therefore the chromosomes get further apart. This is interesting here. It says Okay, so if you said the nucleus expands, you could have got a mark for the second mark, but not chromosomes expand. Okay, so chromosomes are the coiled up DNA. A chromosome can't expand. You can't put water into a chromosome. It is just coiled up DNA. Moving on. 1.4. The dark stain used on the chromosomes binds more to some areas of the chromosomes than others, giving the chromosome a striped appearance. Suggest one way the structure of the chromosome could differ along its length to result in the stain binding more in some areas. Ooh, this is a tough one. I haven't actually looked at this mark scheme yet, so I'm kind of just improvising. So here are my thoughts for this question. Okay, so first of all, we might have in some areas of the chromosome, the chromatin might be more densely packed than others. And in some places it might be slightly more loosely packed. It does say, now this question is one of these list principle questions. So it says suggest one way. My other guess, but I have to pick one, would be that you've got some things like repeating sequences that bind the stain more tightly. So you're like trinucleotide repeats, for example. But I think I'm probably going to go with my first answer, which is about chromatin being more or less tightly packed. So let's see if I get this is right. Sometimes you've got to have a bit of a guess. Okay, well, my two answers were both correct, so I'm pretty pleased with that. So this is the first thing I said was the differences in condensation or supercoiling. Now, I didn't use the phrase supercoiling, I said tightly packed. So I think I would have got the mark for that, but maybe we should just make a mental note that when we're talking about the packaging of chromatin, we should talk about supercoiling or condensation. Some areas of the chromosome are more condensed or are more tightly supercoiled, I could have said. And the other thing I said was maybe differences in base sequences. I talked about trinucleotide repeats, which would fall under that. Difference is in histones, interactions with the histones. Okay, so if you'd kind of referenced this area, spec section 8, about methylation of the DNA or acetylation of the histones, that would have also been accepted. All right, in figure 2, the chromosomes are arranged in homologous pairs. What is a homologous pair of chromosomes? This is a simple definition type thing. A homologous pair of chromosomes contains a maternal chromosome and a paternal chromosome with containing the same genes, I would say. Different alleles, but the same genes. Let's just see what they want as the definition. Two chromosomes that carry the same genes. That's the definition that you should remember. I got there in the end. Two chromosomes that carry the same genes. Don't say the same alleles, because they might have different versions of the gene. You could say same loci. A loci is a place where a gene is. But interestingly, you wouldn't get a mark just for saying a maternal and paternal chromosome. That wouldn't quite do it. A maternal and paternal version of a chromosome that carries the same genes. (1.6) Give two ways in which the arrangement of prokaryotic DNA is different from the arrangement of human DNA in figure 1. Well, it's not in chromosomes, it's not associated with histone proteins, and it's not in a nucleus. 3 things. Now, yeah, I think all those would be marked, because I'm just thinking it says in figure 1, so I think we have to specifically reference a difference that is visible in figure 1. So I presume in figure 1, does it talk about the nucleus? I would probably say chromosomes and histone proteins, I think. You can't tell it's in a nucleus. It doesn't show the nucleus, so I'm gonna say that. Yeah, so my suspicion was correct. It doesn't have anything about in the nucleus or not in the nucleus, because the nucleus is not visible in figure 1. So that's why that kind of thing, that true fact, is not acceptable in this specific instance, which is kind of annoying because it's like an obvious thing that you might have said. So what you could have said, prokaryotic DNA is circular as opposed to linear. That was visible because the chromosomes are long sections of DNA, not circles. So that's visible in the picture. They're not associated with histone proteins. And what else did I say? Only one molecule piece of DNA or present as plasmid. Yeah. Okay, those are the three things you could have said. I actually forgotten what I said in my answer. So I don't know if I would have got 2 marks or just 1. The interesting exam technique point there was that you had to reference something that was visible in figure 1. Okay, so give yourself a mark out of 9 for that question. That question was all about kind of mitosis and chromosome condensation. And I think that was one of the predictions that I think might come up this year, something about chromatin and condensation of chromatin. So that's something that you need to know. Okay, moving on to question 2. It says, the student investigated the effect of surface area on osmosis in cubes of potato. Okay. He cut 2 cubes of potato tissue, each with sides of 35 millimeters in length. Just going to kind of like draw a little cube of potato just so I can kind of visualize it. Each one 35 millimeters in length. He put 1 cube into a concentrated sucrose solution. He cut the other cube into 8 equal size smaller cubes. Okay, that's interesting. So we've got that big cube, and then he's chopped it up into 8, yeah, like this. 1, 2, 3. We get 1, 2, 3, 4, 5, 6, 7, 8. Yeah, that's 8. Okay. He recorded the mass of the cubes at intervals. Okay, so here we can see the total mass, and because this is concentrated sucrose solution, we need to kind of think what's going on here. Maybe annotate the question. Concentrated sucrose solution has a very low water potential, so therefore we're talking about osmosis. We're going to have water leaving these cubes by osmosis. Okay, so that's the gist of the question. Now let's see what they're actually asking. But we can already see, before you even get to the question, we can already see that there's a big difference. The 8 small cubes are losing mass more rapidly than the 1 big cube, and this should be screaming at you surface area or surface area to volume ratio. Okay, describe the method the student would have used to obtain the results in figure 3. Start after all of the cubes of potato have been cut. Also consider variables he should have controlled. There's so much here for 3 marks. There's like masses of stuff you can say. Okay, describe the method. Really, we're talking about record mass in sucrose solution. Remove, blot. Remember, when you take the potato cubes out, you've got to blot them on some tissue paper, not too much, just a few times, to remove the surface water. So you might say that blot to remove the surface water, and then record the mass again, and then calculate the difference. And actually, I would normally do percentage change, but have they done percentage change here? No, they haven't. They've just done the total mass. And actually, in fact, you don't need the difference mark. That's actually not given, because you can see that they don't plot the difference. They're just plotting the actual mass. And they would do this then I suppose they would then put the cubes back in again. Yeah, actually, that's how they do. They'd have to record the mass and then put cube back in again. Okay, because what they're doing, I don't know how often they're recording this, but let's say they record every 20 minutes. So they do this for 20 minutes, they take them out, they record the mass, they put the cubes back in again, they take them out at 40 minutes, they blot, they record the mass, and so on and so on. I presume that's what they do, okay? Because obviously they can't record the mass of the cubes whilst they're actually in the solution. They're going to have to take them out intermittently. Okay, things that are the same, obviously, the control variables. Of course, I can't believe this is only 3 marks, to be honest. I would have thought this would be 6 marks. Control variables is the sucrose solution, because it just says concentrated sucrose solution. What concentration? So the concentration of sucrose solution and the volume of sucrose solution. What am I not saying? What am I not saying? I'm not saying amount. Amount is a bad word, especially when talking about liquids and experiments. Never say amount. It's vague. Are you talking about the concentration or the volume? Amount is a bit, well, I don't know what we're thinking about. So concentration or volume, not amount. The other thing that probably you could have said is temperature, because temperature would affect the rate of osmosis as well. So those are things that you could have said. Okay, hopefully that's all on the mark scheme. Let's quickly look at it and just check that I'm not talking nonsense. Is it a one? Measure mass of stated cubes at intervals. Fine, yep. Method of drying cubes for measuring, blotting, yep. Okay, I didn't give a method of controlling temperature. You have to have a method. Gosh, is this, maybe let me reread the question. I guess you have to describe, they're not separate points. You have to describe how they would keep the method the same. How they would keep the temperature the same. So for this one, I might have only got 2 marks out of 3 here. For this one, probably water bath, maybe is a good one. Put them all in a water bath, or actually just leave them all at room temperature is valid actually. So for this one, this is an interesting one. So for the 8 cubes, you don't want to have them all stuck together with the surfaces touching, because then that kind of eliminates the surface area boost, I suppose. So you want to, like, when you drop all the 8 cubes in, you maybe just want to quickly stir them around so that they're all separated and not clustered together. Time intervals between every 5 minutes with a maximum every 40 minutes. I suggest every 20. So yeah, interesting. Okay, moving on. 2.2. The loss in mass shown in figure 3 is due to osmosis. The rate of osmosis between 0 and 40 minutes is faster in B than in A. Is the rate of osmosis per millimeter squared per minute different between A and B in this time? Okay, so we need to work out the surface area of both cubes. Okay, right. So I'm going to take you through the marking of this. Surface area of the big cube and surface area of the 8 little cubes. Okay, the big cube is 35 millimeters squared times 6. Okay, so the big cube is 35 times 35 millimeters. That's one single face, and the whole thing is times 6 because there's 6 faces of the cube. Is that right? Yeah. So let's do that on my calculator real quick. 35 times 35 equals times 6 equals. Okay, so we've got 7,350 millimeters squared. And on B, okay, so let me just think. So each face here, what's the side length? It's half of 35, which is 17.5. Yeah, 17.5 is the side length of each cube. So this one is 17.5 times 17.5, or 17.5 squared, times 6, and then there's 8 cubes times 8. Okay, so what's that? 17.5 times 17.5 equals times 6 equals times 8 equals. So we've got 14,700 millimeters squared. Okay, and then what's this thing? It says the rate of osmosis per millimeter squared per minute different between A and B during this time. My goodness, there's a lot of maths to do to get these marks. All right, so for A, I'm going to change color. A, the kind of rate of osmosis in the, does it say in the first 40 minutes? It does. So in the first 40 minutes, A has gone from 62, we've got to really pinch in and make sure that we're reading this accurately. A has gone to 57. So 62 to 57, which is a change of 5. Yep, 5. And then for B, we've gone from 62 to 53. So that's another 4, so that's 9. Okay? 5 versus 9. The rate of osmosis for B equals 9. Right, now I almost missed something. You've also, because it wants the rate of osmosis per millimeter squared, you've also got to divide this 5, which is the total like grams of water loss, divided by the 40 minutes. Okay, so 5 divided by 40. So 5 over 40 and 9 over 40. So therefore the rate of osmosis for A, 5 divided by 40, and I get a very long number, which is 0.00001231417. So I could write that as 1.7 times 10 to the minus 5. And this one, 0.225, and this one's going to be divided by 14,700. So 0.225 divided by 14,700. 1, 2, 3, 4. So again, it's 4 zeros, so it's going to be 1.5 times 10 to the minus 5. Okay, so yes, they are different. Not a lot, but a bit. 1.7 times 10 to the minus 5 grams per millimeter squared per minute. Let's put the units on. Grams per millimeter squared per minute. So minus 2. The same thing over here. Grams per millimeter squared per minute. So they're different. So what are the marks for this? Well, let's hope they give you some marks for working. So if you get the calculation of the rate per millimeter squared for both sets of data, then that's 3 marks straight away. So I got 1.7, and what did I get? 1.7 and 1.5. So that's okay. So 1.7 is in the middle there, and 1.5 is in the middle of there. So yeah, I got those. But if you didn't get those two numbers, and you made a mistake somewhere, there's still marks of the working. So if you first mark, you could have got just the areas of the cubes, 7350 and 14,700. Then if you remember to divide by the 40 to get the rate in the first 40 minutes, so somewhere between 0.12 and 1.3, or 0.22 and 0.23, and then this is like an error carried forward mark. So if they can tell that you did do a rate divided by a surface area, even though your rate and the surface area were wrong, you could have still got a mark, which is quite nice, I suppose. That was a really hard mathematical question. I hope you enjoyed it. Okay, moving on to question 3. So it says, bees are flying insects that feed on nectar made in flowers. Many different species of bees. Scientists investigated how biodiversity of bees varied in 3 different habitats during a year. Here's the important stuff. Okay, so 3 different habitats, and they collected bees from 8 sites of each habitat 4 times per year. This looks interesting. Okay, 8 sites of each habitat 4 times per year for 3 years. Okay, so that kind of gives us an idea of the number of repeats. The scientists' results are shown in figure 4 in the form they presented them. So we've got habitats, we've got natural, town, and farmland. And over here, this is the number, the mean number of bees collected at different times throughout the year from the 8 different sites, I guess. And then this is the bee species richness. So species richness, in case you didn't know, you need to know what that means to interpret this. So species richness is the number of different species of bee, okay, present. Number of species. Okay, that's the first question. What is meant by species richness? So that's just the number of different species of bee they found. Let's check the mark scheme, just check that that is the exact definition, or if we need a better definition. The number of species in a community. Probably, if I'd written this down, I probably would have said in the area, so I might have got that mark, but let's be really exact and let's memorize this definition. The number of different species in a community. It would have accepted ecosystem if you said that. It's probably the best, next best answer. Number of different species in a community or in an ecosystem. (3.2) From the data in figure 4, a student made the following conclusions. The natural habitat is most favourable for bees. The town is the least favourable for bees. Do the data in figure 4 support these conclusions? Explain your answer. So what is this? It's 4 marks, and it's kind of nice that they've broken it down into 2 parts. So what we're looking at here is we essentially need to have a statement that supports number 1, the natural habitat is most favourable for bees, and a statement against number 1. You need a statement that supports number 2 and a statement against number 2. So let's find some statements. First of all, let's find some information that says the natural habitat is most favourable for bees. Favourable for bees. No difficulty speaking today. Okay, so the natural habitat is most favourable for bees. Okay, well here's some good information. This one. So I'll just kind of label this as 1 plus because it supports statement 1. So what we can see here is the natural habitat has the greatest species richness of any of the habitats all throughout the year. So that's really clear. Always has the highest species richness. What about something that doesn't support statement 1, that the natural habitat is best? Well, how about this bit of information here? I'll highlight with maybe this kind of pinkish colour. This kind of information here. Okay, in the winter, it looks like, yeah, later on in the year. So this is, I'm going to label this as 1 minus. So what we would say here is in the winter, the mean number of bees collected in the natural habitat is lower than the other habitats. So it looks like the farmland habitat has the greater number of bees after around, and we need to be precise with our answer, after around day 200. So that's how I would phrase it. So after day 200 of the year, the farmland habitat has a greater mean number of bees collected than the natural habitat. Okay. All right, let's go on to statement 2 and look for some evidence for and against. The town is least favourable for the bees. Let's find some evidence. The town is least favourable for the bees. Okay, so evidence that says the town is least favourable. Okay, so I'd probably pick this bit over here, this bit of the line here. So I'd say for most of the year, for over half of the year, the town has the lowest species richness. And if we wanted to be precise, we'd say after about day 130, so post day 130, the town has the least species richness. But then that's statement 2 supporting the fact that the town's not great. Is there something that we can use to argue against that? Well, we could also, well, I guess we could equally talk about the fact that the species richness in the town is higher than farmland for almost half the year. Or we could maybe talk about this bit here. Maybe we talk about this bit here. And we talk about up until about day 150, the town had a greater number of bees collected on average compared to the farmland. So I'd probably say 2 minus. Really, it's just about looking at the data and the graph and trying to like pick out something to support your argument. Let's look at the mark scheme and see all the different things that you could have said. There's so much. Right, okay, so I'm not going to read through all of these. You can kind of pause and kind of look for yourself, but natural is best. There are 1, 2, 3 things you could have said. Natural, and then sort of to go against that argument, no, natural is not best. This. And then for the second kind of part of that question, supporting the idea that the town is bad, you could have said this or this, and going against the idea that the town is bad, you could have said those. Okay. Oh, interesting general stuff at the bottom. Pause there if you want to look at that mark scheme in more detail, but I'm going to pan down. Okay, I'm going to pan down. Interesting general stuff here. Generally no, because we actually just looked at species richness, but we didn't measure an index of biodiversity. So something like Simpson's index, that takes into account two different things. It takes into account species richness and also species evenness, which is how kind of even the different species are. You could have like 10 species, but almost all of the bees are just one species and the others are very few. That would be an uneven distribution across the different species. Species evenness means there's about the same number, more or less, of each of the different species that are there. And Simpson's diversity index takes that into an account. Okay, let's keep going. 3.3. The scientists collected bees using a method that was ethical and allowed them to identify accurately the species to which each belonged. In each case, suggest one consideration the scientists had taken into account to make sure their method was ethical. Okay, so ethical bees, so basically the bees aren't harmed, or something like that, and are returned alive back into the environment. That's ethical. You're not mistreating the living things. 2, allow them to identify accurately the species to which each belonged. I presume this would be something like use an identification key. You can get these like charts of different bee types. I actually tried to do this last summer in my parents' house. I had a bit of fun trying to spot how many different bee species I could find. I think I found about 4 or 5. So some identification chart probably is what they're looking for. Let's just look at the mark scheme, see if those 2 points are the ones they're looking for. Yep, okay, so must not harm the bees, that's the ethical thing, or allow the bee to be released unchanged. This is the ethics. And the second part, yeah, I would think I would have got the mark. A key is a good word to use. Now, technically, a key is one of those kind of flow diagrams that says, is the bee yellow, yes or no? If no, is the bee orange or black, yes or no? If it's orange, does it have parts of black or is it fully orange, yes or no? And you kind of go through that process, yes or no questions, and it gives you what bee you've got. Okay, but it would have said except photographs or specimens to identify species. Okay, here's a picture of a bee chart. Go have some fun, identify some bees. 3.4. Suggest and explain 2 ways in which the scientists could have improved the method used for data collection in their investigation. Okay, so for this, always when we got this kind of suggest and explain, you kind of want to go back to the beginning of the question and reread how they did their data collection. So, all right, let's go back up here. And it's really going to be the stuff that we highlighted up here. Okay, so they used 8 sites 4 times per year for 3 years. So they perhaps could have done 8 sites from each different habitat. So they could have used more sites per habitat, maybe 20 sites per habitat, and they could have also used, they could have sampled on more than 4 times per year, or they could have sampled for a greater number of years. Now for each one you have to explain why it'd be better, and essentially it's going to enable you to get more data and to get a more accurate average by removing anomalies. That's kind of what I'm guessing. But I'm going to look at the mark scheme to see exactly what they're looking for the explanation, because I'm not entirely sure what they want. So collect at more times of the year. Counted, yeah, collect from more sites or for more years. So these are the 2 things that I suggested. This one I didn't spot actually. Now, what was the explanation for this? To allow, yeah, increase the accuracy of the mean by identifying anomalies. That's what I said. And more points on the graph. Interesting. Well, there are no points on the graph. This middle one counted the number of individuals in each species, so they could calculate the index of diversity. That is a good point there. So what we're saying is they just counted the number of species, but we've got no information about the species evenness. We don't know the numbers per species, so potentially that could be somehow quantified and put on the graph. It would be better information. Let's move on. Okay, 3 of the bee species collected in the farmland area were Peponapis pruinosa, Andrena chlorogaster, and Andrena piperi. Okay, this is interesting. What do the names suggest about the evolutionary relationship between these bee species? Explain your answer. Okay, well, these are binomial names. Okay, so what we have in a binomial name is we have the genus, which has always got a capital, and then we've got the species. So genus and species. So 2 of these belong to the same genus, and one is in a different genus. So what we've probably got, sort of a phylogenetic tree, if I draw it, it's difficult to say exactly how it's going to look, but we've got 2 bee species that are kind of relatively closely related. I'm just going to kind of abbreviate by Andrena chlorogaster, and here we've got Andrena piperi. Like so, those are more closely related, and then somewhere less closely related is this one, Peponapis pruinosa. Okay, so that's kind of the relationship there, and you want to use these words genus, species, and maybe binomial name in your answer. 2 marks, should be plenty of things to say there to get the 2 marks. Let's see the mark scheme. So you need to mention that A. chlorogaster and A. piperi are more closely related because they're in the same genus. I think that word genus is needed, and this sort of idea of close relationship. So it must be a comparative statement. So this is interesting. So you can't just say A. chlorogaster and A. piperi are closely related because they are in the same genus. That would get you 1 mark. You have to say that they are more closely related than to the other one, P. pruinosa, because it's in a separate genus. Okay, let's move on. Okay, question 4. Formation of an enzyme substrate complex increases the rate of reaction. Question 4. Formation of an enzyme substrate complex increases the rate of reaction. Explain how. Okay, great. This is easy marks. Hopefully we all got this. What do we need to say? We need to say the substrate fits into the active site. During that time when the substrate is in the active site, they are complementary, when you say the word complementary, and interactions between the amino acids in the active site, they stabilize a transition state of the substrate molecule, resulting in a lower activation energy so that the substrate can be broken down more quickly. Okay, so the keywords we're looking for are complementary, active site, what else? Activation energy. That should do it. Let's look at the mark scheme. Ooh, this is interesting. Reduces activation energy, that's there, due to bending bonds. Gosh, I've never seen that before, but then I'm kind of new to AQA marking, so this is what they want you to say. Due to bending bonds. Or, without enzyme, very few substrates have sufficient energy for the reaction. Let me see what else it accepts here. Due to stress, pressure, tension on bonds, or due to weakening bonds. Okay, so here's a little clip from a protein data bank video that I found online, which kind of shows this. So this is a substrate going into the active site, and what we can see here, the kind of amino acid on the right, which I think is histidine there, which have the blue atoms, it is interacting with the substrate molecule, and you can kind of see the atoms in the amino acids which surround the substrate are constantly kind of tugging or pulling at the substrate. So this is what it means by bending bonds. Really, there are interactions, and in this particular reaction, if you do chemistry, there's I think electrophilic addition and nucleophilic addition and things like that going on. But the key point is that the enzyme is unchanged at the end. Okay, so let's go back. Okay, 4.2. Scientists measure the rate of removal of amino acids from a polypeptide with and without an enzyme present. With the enzyme present, 578 amino acids were released per second. With the enzyme. Without the enzyme, 3 times 10 to the minus 9 were released per second. Oh god, it's just a standard form thing. By how many times the rate of reaction is greater with the enzyme present, give your answer in standard form. Okay, so what we're doing is looking at 578, which is the rate now, divided by the rate before, which is 3.0 times 10 to the minus 9. And I presume you can probably just do this in your calculator if you have a scientific calculator, which I do not. So I'm going to do this slightly long-winded. So 508 divided by 0.1, 2, 3, 4, 5, 6, 7, 8. And there's the 9. So I'm going to do that. It's a big number. So it's 1.92. Well, actually, no, 1.93 times 10 to the times 10 to 11. Is that right? Yes, it is. 1.93 times 10 to the 11. You can get 1 mark for the calculation. A 1 mark for just kind of getting the standard form wrong. So having 1.93 times 10 to the something, but getting it wrong. Okay. Moving on. Another scientist investigated an enzyme that catalyzes the following reaction. Okay, this is hydrolysis of ATP. The scientist set up 2 experiments, C and L. Experiment C used the enzyme and different concentrations of ATP. Okay, experiment L used the enzyme, different concentrations of ATP, and a sugar called lixose. Okay, so we're just looking at how lixose affects this enzyme, I suppose. Scientists measure the rate of reaction in each experiment. Results are shown in figure 5. So it looks like the lixose, that's how you say it, is increasing the rate of reaction at all concentrations of ATP, it would seem. Right, what are the questions? Calculate the rate of reaction of the enzyme. Calculate the rate of reaction of the enzyme activity with no lixose at 2.5 millimoles per dm cubed of ATP as a percentage of the maximum rate with lixose. Calculate the rate of reaction of enzyme activity. This is such a ridiculous question. This is just designed to trip you up. Enzyme rate of reaction, no lixose at 2.5 moles versus maximum rate shown with lixose. God, what a silly question. Okay, so no lixose at 2.5 moles. Let's find that from the graph. Right, no lixose at 2.5 moles. This is 2.5 moles, so we're looking for this point right here. And then, okay, there's a further complication because you might misread this. You might misread it as 4.1, but actually that's 4.2, 0.4, 0.6, 0.8. Actually, hang on a minute. No, that's 4.4. Good thing I checked. That is 0.44 because each of these increments, 1, 2, 3, 4, 5, is worth 0.4. So that's 0.44. And then the maximum rate is at 1.4. There, that's the maximum rate. So that's 0.44. And this is, did I say 1.4? Yes, I did, 1.4. Calculate the rate of reaction of the enzyme activity as a percentage of the maximum rate. So basically what 0.44 is what percent of 1.4. So then we just do the calculation. I'm doing red. So it's 0.44 divided by 1.4. And we times it by 100 to make it percent. Let's do that. 0.44 divided by 1.4 equals 0.31. Or 31%. 2 marks, hopefully. Check mark scheme. Does it say to how many significant figures? It doesn't say to how many significant figures. I wrote 31. Would I get a mark? Except any number of significant figures greater than or equal to 2. So 31 is okay. So 31.4 or 31 is fine. These numbers are slightly off. Yeah, so like you could have got these numbers and got a mark if you'd made some of those mistakes that we highlighted. So for example, if you'd incorrectly read 4.2 and 1.4, etc. like that. Okay, let's move on. 4.4. Lixose binds to the enzyme. Suggest a reason for the difference in the results shown in figure 5 with and without the lixose. Okay, well this is kind of interesting because it's almost like the opposite of an inhibitor. So lixose is somehow improving the activity of the enzyme. So what could it be? So maybe, actually I'll draw a little diagram of kind of what it would look like. It's actually called an allosteric activator. So essentially this is the enzyme, and I don't know, here's the active site, something, yeah? And the lixose molecule maybe kind of binds over here, potentially, and when it binds, it just subtly changes the shape of the active site somehow, such that the substrate molecule then kind of can fit better in the active site. Okay, so we're getting a better fit. And that's what we need to say, but let's just look at the mark scheme and see if that's how we're supposed to say it. Okay, so we need to talk about tertiary structure. I made a mistake in my explanation. I said the word shape, and you really shouldn't use the word shape when talking about enzymes or proteins. You should always say tertiary structure. So binding alters the tertiary structure. This causes the active site, and that's underlined. You need that to change shape. And then you always follow that up when talking about enzymes with more enzyme substrate complexes form per minute or per second or whatever. They form more quickly, or I guess you could have also said, look down here, it further lowers the activation energy. It lowers it even more than normal. Yeah. Okay. Let's move on. Okay. Draw the general structure of an amino acid. This is an easy mark. Hopefully everyone knows this. Central carbon. On the one side, we've got this carboxylic acid group. On the other side, we've got an amino group. And we've got a hydrogen and an R group. It shouldn't matter which way you put those two round. It doesn't matter. Okay, 5. There's got a table here. Table 1 shows mRNA codons and the amino acid coded for by each codon. It also shows some properties of the R group of each acid. Okay, so we've got some uncharged ones, positively charged ones, and negatively charged ones. Okay. The genetic code is described as degenerate. What is meant by this? Use an example from table 1 to illustrate your answer. So degenerate means multiple codons can code for the same amino acid. And an example, let's pick, well, proline, for example. Proline is very degenerate. There are many codes for proline. What could code for proline? Well, you could have CCU would code for proline, CCU, or you could also have CCC, that's this one here, or CCA or CCG. So there are 4 degenerate codons that all code for the amino acid proline. Okay, let's check the exact wording of the mark scheme. There's the amino acid. So more than one codon codes for a single amino acid, and giving a suitable example. For example, the proline one that we gave. A scientist investigated changes in the amino acid sequence of a human enzyme resulting from mutations. All these amino acid changes result from the single base substitution mutations. This enzyme is a polypeptide 465 amino acids long. Table 2 shows the results of 3 of the base substitutions. Okay, amino acid number. Okay, so 3 different mutations. What is the minimum number of bases? Strange. Oh, okay, I understand now. What is the minimum number of bases in the gene coding for this polypeptide? Right, so it's 465 amino acids long. That's what we need to know. And there are, each amino acid is coded for by a codon, which is 3 bases long. So it's times 3 to get the answer. Now, it's only 1 mark, so I think that is the correct answer, 465 times 3. If it... I doubt that it will be 1,000. It could be. There's a small chance, and I don't really know what the answer will be, that it might be 3 more because you might include the stop codon, but I don't think so. So let's just let's just have a look at the mark scheme, see what they say. 1395, yeah. Ah, look, it does accept. If you wrote this, you could have added three more on there, or 6 more, in fact, for the start and stop codon. So if you had said 1, 3, 9, 8, that would have been with the stop codon, or maybe 1401 for a start and a stop codon. So those would have been accepted. That's nice of them to do that. So I'll put +3 or +6, depending on whether you talked about the start and stop codons. Okay, 5.4. Use information from Table 1 to tick 1 box that shows a single base substitution mutation in DNA that would result in a change from valine to alanine at amino acid number 203. Okay, this is just reading the table, so we need to go and find what valine, the code for valine is. Valine. So valine is GU and then something. GU, and then it could be any of those. GU something. So which one's a valine down here? It's going to be a GU, that's a valine, and that's a valine. That's not a valine, that's not a valine. So those are out. What about changing to alanine? What's the code for alanine? We go back up, we look at the code for alanine. Alanine is GC something, GC something. So we go down and we try and find GC something. This is it, GCA. Okay, so this is the answer there. Okay, a change from glutamate to lysine, glue to lyse, amino acid 300 had no effect on the rate of reaction catalyzed by the enzyme, whereas the same change at amino acid 279 significantly reduced the rate of reaction catalyzed by the enzyme. Okay, use all the information in your knowledge of protein structure to suggest reasons for the differences between the effect of these 2 changes. Okay, well, there's 3 marks here. First, I think we want to talk a little bit about what this glutamate to lysine change is. So let's go and look at the table again, glutamate to lysine. Now, glue, where is glue? Okay, so glue is a negatively charged. Okay, is negatively charged, and lysine is positively charged. That's interesting. So glue negatively charged, lysine positively charged. Definitely going to be a mark for talking about that. So that's negative, and that's positive. So this is going to affect, so mention that, this is going to affect potentially ionic interactions within the tertiary structure of the enzyme, but possibly one of these amino acids is maybe in the active site of the enzyme, and one of them isn't. So possibly the amino acid 279 is in the active site, which makes the active site differently charged, or means it does not bind to the substrate anymore, whereas in this one it doesn't have an effect on the active site. Let's check the mark scheme and see the exact terminology that you need to use. Okay, so the first mark, as predicted, is talking about this negatively charged to a positively charged change in the amino acid. The fact that amino acid 300 does not change the shape of the active site, whereas if we change the amino acid 279, it probably does change the active site. But you do need to talk about, okay, so you need to talk about the type of bonds that may change the active site. So you need to, in the sort of same thought. So for amino acid 279, this swap from positive to negative may affect a ionic bond or a hydrogen bond or a disulfide bond. And so the shape of the active site changes. I actually don't think it would really affect a disulfide bond because that's only if you have the amino acid cysteine. Cysteine not involved, so I don't really see why it would affect a disulfide bond, but never mind. Okay, let's keep going. Okay, moving on to question 6. Figure 6 shows a faulty form of meiosis that can occur in some plants. So here we have the original cell chromosome content of a diploid parent cell. So notice here we've got 2 pairs of chromosomes, okay, because we can see we've got a long one, and another long one, and then we've got a short one, and another short one, like that. Okay, after the first division of meiosis, we've separated out the homologous pairs. So here we have this is haploid. I'm just kind of annotating as I go to kind of follow along, and this is also haploid. The chromosomes are still that kind of X shape because they've been replicated earlier on. Okay, so the second division of meiosis, 2 daughter cells do not develop. Okay, interesting. But we basically just seemingly kind of pull apart those pairs there. Okay, interesting. Chromosome content of the 2 daughter cells that develop as gametes. Diploid 2N. I guess we call them diploid. I'm finding this a bit weird to call this diploid because you know, both of these long ones will be genetically identical and both of these short ones will be genetically identical. I guess they do still have 2 copies of each chromosome. It's just that the copies are identical. So it's kind of a diploid, but not really. Perhaps there'll be questions about this as we keep going. Complete figure 7 to show the chromosome content of the cells that would result from a normal meiotic division of the diploid parent cell shown in figure 6. Okay, so after the first division of meiosis, we've got that's exactly, you know, it's like basically we just want to take this whole image and copy it. I'm going to do that now, like so. But what would it look like after? Oh, what would it look like after? Well, each one would have, you know, short, long, short, long, short, long, and short, long. Doesn't matter the order. Okay, so just like that. Let's check the mark scheme. Double check. Check that's what they want from us. Yeah, 1 long and 1 short chromosome, each made up of 2 chromosid held in each. Okay, in the first division. Okay, and then one long and 1 short chromosomes. Yeah. Okay, so this is basically the exact same as before, and this is what we drew. Okay. It says ignore drawing of the centromere. For a second there, I thought we had to put these in. Probably a good idea that I should have done anyway. Okay, 6.2. If 2 diploid 2N gametes fuse at fertilization, it can result in a tetraploid plant which has 4 copies of each chromosome. Okay, red clover is a plant grown to produce cattle feed. Tetraploid red clover plants produce a high yield than diploid red clover plants. Interesting. Whether a red clover plant produces 2N gametes is genetically controlled. Scientists investigated the possibility of breeding red clover plants that only produced 2N gametes. In breeding cycle 0, they grew red clover plants on identified plants that produced 2N gametes. In breeding cycle 1, they used the plants producing 2N gametes to produce offspring. In breeding cycles 2 and 3, they identified plants producing 2N gametes and used these to produce offspring. So this is essentially artificial selection. So breeding cycle 0, 1, 2, 3. Number of plants that did not produce 2N gametes is going down and down and down. And number of plants that did produce 2N gametes is going up. The scientists used the following null hypothesis. The proportion of plants that produces 2N gametes will not change from one breeding cycle to the next. Ah, yes, I remember this question. I have seen this one before. Complete table 3 to show the expected number of plants that did not produce 2N gametes and the expected number of plants that did produce 2N gametes after each cycle. Now, the key thing here is the wording of the null hypothesis. The proportion of plants that produce 2 gametes will not change. The proportion, not the number, but the proportion. So what we've got here is a 50-4 kind of ratio. So, you know, this is 50, essentially 50 out of 54, and this is 4 out of 54, okay? Like as a percentage or as a fraction. So what I need to do is kind of follow that proportion down, but with the different numbers. And then same thing for here. So this is 14 and 42. So 14 and 42 is, what's that, 56. So I need to do the maths I need to do. Gosh, this is annoying. 56 times 50 divided by 54. Okay, what is that? 56 times 50 divided by 54 equals, it's 50, or 51.8. I presume we round to the nearest whole number, so I guess that's 52. And if that's 52, remember this was 56, so that's going to be 4 there. We've got to keep this thing going, so the total number of things here is 46. So I do 50 divided by 54 equals times 46, which is going to be 43. And then over here, we've got 46 total, so there's going to be 3 there. And then down here is 56, so 50 divided by 54 equals times 56 equals 52, and that's the 4. Okay, it's the proportion of plants that won't change, not the number. Let's double check that. $52 and 4. Did I not need to do it for all of those? Oh yeah, I just needed to do it for this top one. Oh, okay. Well, there's a bit of extra work, but yeah, that's correct. Tick. Right, moving on. Okay, so this next one is about chi-squared, 6.3. The scientists tested their null hypothesis using the chi-squared statistical test. So this is a test where you are looking at your observed frequencies and your expected frequencies, and you're seeing whether they are different enough to be significant. After one cycle, their calculated chi-squared value was 350. That is quite high. So the critical value at p0.05 is 3.841. So this means it's very significant. Okay, so this means that the difference, this number, so you have to say that the calculated value of chi-squared was far bigger than the critical value. So therefore, the difference between the observed and expected frequencies is significant. Like furthermore, we can say that it's highly significant because it's so much bigger than 3.84. Let's check the mark scheme, see what the wording is looking for. Oh, okay. Oh, difference is significant is not accepted. Okay, I stand corrected. Other examples like that. So what does AQA want? AQA wants you to say that you reject the null hypothesis. So okay, let me learn this, let you learn this. So if the calculated value of chi is bigger than the critical value, we reject the null hypothesis, and therefore there is a less than 5% probability that the difference occurred by chance. I think this is something to remember. The probability that the difference occurred by chance is less than 5%. So we can conclude, therefore, that the proportion of so we can conclude, therefore, that the proportion of plants that produce 2 gametes does change from one breeding cycle to the next. I need to get that wording in my head. We reject the null hypothesis. There is less than 5% chance that the difference was due to chance. Use your knowledge of directional selection to explain the results shown in Table 3. Well, essentially, we are doing directional selection. Humans are the selective pressure. So there's a strong selective pressure that causes a change in the allele frequency. I think those are the words that we need. Yeah, so we need to talk about the frequency of alleles increased. The scientists, the scientists were carrying out artificial selection. So the scientists selected the plants that produced 2N gametes, so only these plants passed on their alleles, and therefore the frequency of alleles changed. This is called artificial selection, where humans take the place of the kind of natural process. Okay, moving on to question 7. When a person is bitten by a venomous snake, the snake injects a toxin into the person. Antivenom is injected as a treatment. Antivenom contains antibodies against the snake toxin. This is an example of passive immunity. Explain how the treatment with antivenom works and why it is essential to use passive immunity rather than active immunity. Okay, well, it works. Basically, the antibodies, I don't know, I'm just going to draw V for venom. Here's the venom molecule. The antibodies essentially bind to the venom molecule and block its activity somehow. Okay, the venom molecule is presumably like a protein, probably a protein. So it's just going to block the activity of that protein. Sometimes venom is an enzyme, so it might block the enzyme. So that's how it works. This is weird phrasing. Why is it essential to use passive immunity? Well, basically you can deliver antibodies quickly. So these are antibodies that have been made previously. They're often made in a horse, incidentally. So these antibodies, you've got a large stock of antibodies in a needle, and they can be injected into the person soon after they've been bitten by a snake. So we can rapidly neutralize the venom. If it was active immunity, the processes of, you might say, the processes of clonal selection and clonal expansion and the production of antibodies by B cells or beta lymphocytes takes a lot of time, and the person would be dead by then because snake bites are serious things. So yeah, okay, so that's it. So antibodies bind to the toxin and cause its destruction. Okay, weird. This is, I got this right. Active immunity would be too slow. But for the first point, they want you to say that the antibodies cause its destruction. For bind, accept, attach, ignore, attack, bind to the toxin and cause its destruction. You could say it binds to the toxin and causes it to agglutinate, that means stick together, but it does not allow neutralize. I would have probably said blocking its activity, but I don't think that would have been given the mark. Don't know why, to be honest, but that's the way it is. Okay, let's keep going. Okay, here's a procedure used to produce antivenom. Okay, you get the snake, you get the venom, use some snake milking, basically get them to bite like a kind of cup, and the venom goes into the cup. The preparation of venom mixtures, preparation of venoms for use as vaccine. In a parallel stream, you get some animals and you quarantine them and observe by a vet. Often this could be a horse, by the way. And then you vaccinate the animal. So when you vaccinate the animal, by the way, you don't just use an enormous quantity of venom straight away. Typically what happens is you vaccinate the animal with a small quantity of venom, and then you wait a few weeks, and then you do a little bit bigger quantity of venom, and then a little bit bigger quantity of venom, building up the tolerance. So you collect the blood from the animal. So this is the horse. The vet keeps an eye on it, and then eventually, actually after you build up the tolerance, then the horse will be producing large quantities of antibodies in the blood. So then we can kind of really extract a fair amount of antibodies from the blood. 7.2, a mixture of venoms from several snakes of the same species is used. Okay, so I guess this means that there may be some this is suggesting that there may be some variability in the venoms from different snakes, members of the same species. It would be no good if your antivenom only worked on, I don't know, 20% of king cobra bites. Okay, you want it to work on all king cobra bites. So use a range of different venoms from different king cobras to kind of to ensure that the antibodies can bind to the different diversity of proteins within king cobra antivenom from different populations of king cobras, I suppose. Let's check the mark scheme for that, see what it says. Maybe different form of antigen or toxin, different mutations or alleles. So different antibodies need to be complementary to several different antigens. Yeah. Okay, I think I probably would have said that. I'll just pause there if you need to, and let's move on. And then it does finally talk about horses. Horses or rabbits can be used to produce antivenoms. When taking blood to extract antibody, 13 centimeters cubed of blood is collected per kilogram of the animal's body mass. The mean mass of the horse is 350, and the rabbit is 2 kg. Okay, well, this is just saying, why is it better? Well, how much blood can you take from a horse? Okay, it's 13 cm cubed per kilogram, so times 350. That is going to be how much blood can you take from one horse? 13 times 350. It's 4,550 cm cubed, or around 4.5 liters. Whereas from the rabbit, well, you've got 2 times 13, which is 26 centimeters. Okay, so that's not a lot. So basically, you can get a lot more blood from the horse, which means you can produce more antibodies. So it's going to be better. It says use a calculation, so we did that. So horses, because more antibodies could be collected, and then this is the calculation that you need to do. (7.4) During the procedure shown in figure 8, the animals are undergoing observation by a vet. Suggests the reason why. Well, you don't want them to die, basically. Okay, so this is kind of like an ethical type thing. So yeah, kind of this bit, and also this bit. You want to make sure that the horses or the rabbits are not under any duress. So it's kind of an ethical issue. Then there's no harm coming to them. Yeah, so here are the things you could have said. So the animal does not suffer from the venom, vaccine, or toxin. So it does not suffer anemia from the blood collection. Fair point. And also, so the animal does not have a pathogen that could be transferred to humans. That's why they do that quarantine initially. If there was some sort of virus that could pass from a rabbit to a human, then potentially in your antivenom you might also be giving people rabbit virus. Which would be obviously potentially devastating, might unleash a new pandemic. So you don't want to do that. Let's move on. (7.5) During vaccination, each animal is initially injected with a small volume of venom. 2 weeks later is injected with a larger volume of venom. Use your knowledge of the humoral immune response to explain this vaccination program. Okay, so the image that you want in your head is this kind of like primary and secondary immune response kind of thing. So the first time you're exposed to an antigen, you produce a smaller quantity of antibodies more slowly. And the second time you're exposed to that same antigen, you produce a greater concentration of antibodies, and you also produce them more quickly. In addition, they actually fit better as well. So this is why we do this, because the second injection produces that greater concentration of antibodies in the blood that we want to extract. Okay, and actually probably in real life, they probably do this more than twice. They might do it 5 or 6 times over a period of several months, until the horse is this, just pumping out lots and lots of high quality antibodies for this venom. let's look at the mark scheme. So the second dose produces antibodies in higher concentration and more quickly. So this idea of primary and secondary immune response. Now, I don't know if I said this enough clearly. So the B cells specific to the venom reproduce by mitosis, and then plasma cells and memory cells are produced. It says except clone for reproduce by mitosis, because this is, yeah, okay, except this clonal selection. So this is, I used to teach OCR, as you might know. So this is what OCR wants you to say, clonal selection. So clonal selection is the process by which the B cell that is complementary to the venom divides by mitosis. So I probably would have said clonal selection. You can say that, you would be given a mark. Or you could say the B cells that are specific to the venom reproduce by mitosis, brackets, this is called clonal selection, so that when they encounter the antigen again, the second dose produces antibodies at a higher concentration and more quickly. So sometimes, I don't know if you get these graphs in AQA or not, to be honest, but I'll just, this graph here that I kind of drew, this is time, and this is antibody concentration. And what's important is that, you know, the peak here is much higher than the peak here, but also the gradient of this line is really steep, showing that they are produced fast compared to the gradient of this line, which is less steep, which shows the first time round the antibodies are produced more slowly because of that B cells divided by mitosis clonal selection thing. Okay, on to question 8. Scientists investigated the effect of heat treatment on mass transport in barley. They applied steam to one short section of a leaf of the heat-treated plant. Okay, this area is shown by the arrows in figure 9. Okay, all right, I can see them down here. There's an arrow, and there's an arrow. Okay, and then over here, not heat-treated. They did not apply steam to the leaves of the plants. They then supplied carbon dioxide containing radioactively labeled carbon to each plant in the area shown by the rectangular boxes. Okay, so I'm just going to kind of start to annotate with what I think is going to happen. The radioactive carbon, that's CO2, that's obviously going to be taken in by, Rubisco, and ultimately it's going to become glucose, which is going to be radioactive glucose. After 4 hours, they found the position of the radioactively labeled carbon at each plant. These results are shown in figure 9. Okay, so 4 hours later. Okay, I got you. So it looks like, if we zoom in a little, it looks like over here, like there's a lot of radioactive carbon in the leaf where photosynthesis was happening, where carbon fixation was happening, and it's moved down, but it hasn't moved past where the steam was treated, where the plant was treated with steam. That's kind of interesting. Whereas in this one, it's moved everywhere. The radioactive carbon has kind of moved all throughout the plant. So this movement of radioactive sugar is, of course, translocation. It's going to happen in the phloem. So it looks like the phloem cells are sort of killed here somehow by the steam, and that means that translocation can't go beyond where the phloem cells have been killed, whereas here it can. Okay, so that's what it looks like. Right, after 4 hours they found position, blah blah blah. Yeah, we've talked about that. They also recorded the water content of the parts of the leaf that were supplied with the radioactively labeled carbon dioxide. The results are shown in table 4. Okay, interesting. So the water content of the leaf is relatively similar. Okay, it's a slightly higher in the control plant, not heat treated. But if we look at the standard deviation, okay, plus or minus 11, we haven't even got to the questions yet. This is just thinking. So 84 plus 11 is around, you know, I'm not doing the exact math, but it's around 95-ish, and 84 minus 11 is going to be around 73-ish. Okay, so that's, you know, the plus or minus the standard deviation. This one up here is going to be kind of up to like 100-ish, or down to, what's that? That's going to be like 84 or so. Okay, so that's quite a big range. So it looks like there's not really a massive difference between those two plants. Okay, let's get to the questions. 8.1. The scientists concluded that this heat treatment damaged the phloem. Yeah, I think that's fair. Okay, how do the results shown in Figure 9 support this conclusion? Well, what I would say is that in the heat-treated plant, radioactively labeled sugar does not pass the point where the heat treatment was applied, whereas in the non-heat treated plant, radioactively labeled sugar has moved from the leaf all throughout the plant and is found, you know, even in the roots of the plant. It has been moved by translocation in the phloem. We could say that the phloem is the tissue in which translocation occurs. Let's look at the mark scheme and see what it says. Okay. Okay, so this one, point 2, this is what I said, mass flow or translocation in the phloem has happened throughout the plant, only in the plants that were untreated, whereas it doesn't get past the treated point in the other plant. And then I kind of mentioned this, that heat treatment damages living cells. So transport in the phloem throughout the plant is only in plants is stopped or blocked at that point where it's been heat treated. Things that we probably are also worth saying, which I mentioned but then I didn't maybe put in my answer, is that the radioactive labeled carbon is converted into sugar during photosynthesis in the leaves. We can also say that the phloem requires living tissue or requires living cells, and so on. Okay, let's look at 8.2. Okay, the scientists also concluded that this heat treatment did not affect the xylem. So yeah, again, this is because the water content is not very different. So, and we also know that the xylem are kind of dead tissue. So these are things we can say, that the xylem are mainly dead tissue. They're hollow tubes, they're lignified, there's no active transport required in that tissue. And the water content in the treated and untreated leaves, although a little bit different, the standard deviations overlap, so they don't appear to be significantly different. So let's look at the mark scheme. The water content of the leaves was not different because the standard deviations overlap, or the difference is not significant. Water is still being transported in the xylem. Movement in the xylem is passive, so unaffected by heat treatment. I think I didn't use that phraseology. So movement in the xylem is passive. Yeah, movement in the xylem is passive. Okay, let's go onwards. The scientists then investigated the movement of iron ions, Fe3+, from the soil to old and young leaves of heat-treated barley plants and to leaves of plants that were not heat-treated. Movement of iron ions, Fe3+. Okay, heat treatment was applied halfway up the leaves. From soil to, okay. The scientists determined the concentration of Fe3+ in the top and the lower halves of the leaves of each plant. Strange. Ratio, and this is the, oh my god, data. Ratio of Fe3+ concentration in the top half of leaf to that in the lower half of the leaf. Okay, so the first thing to notice is the top half has less Fe3+ than the lower half, in any case, because the ratio, this is kind of maxing out at sort of 0.45-ish. But in old leaves, the heat treatment doesn't have much effect. Okay, maybe a little effect, but we don't know if it's significant, but it doesn't look that big of an effect. Whereas in the young leaf, it really does have an effect. Okay, so for some reason, in the young leaf, heat treatment halfway up stops ions being able to get up the leaf. I wonder why that is, I'm not sure. Let's keep going and find out what the question is. Okay, what can you conclude about the movement of Fe3+ in barley plants? Oh boy. Oh man. Use all the information provided. This is a hard question. I don't know if I'd get full marks for this, but I'll try and get some marks that I can. Okay, so what can we conclude? It says use all the information. So I presume we can use like information from this figure and also previous figures. But what can we conclude about this? So I would say that this no effect here, what does that mean? Well, that means in old leaves, Fe3+ movement is passive, and potentially in the xylem vessels, because we already talked about the fact that xylem vessels are unaffected by the heat treatment because the water content of the leaves was not different. Okay, so we could mention that. That's one conclusion. But it would also follow that in young leaves, the Fe3+ movement is different. It must be active. It must be an active process that requires ATP or requires living tissue. And maybe is in the phloem. Okay, could be. So that's a bit of a guess. I'm wondering why might that be? I think a young leaf that's actively growing, yeah, is probably a net sink for sugar because it's taking the sugar in from other leaves, so pulling the sugar in from the phloem to grow, to build the new leaf, and only once the leaf is fully formed and kind of old, then it's doing its maximum rate of photosynthesis, and then it is a net contributor of sugar to the plant. So then kind of the xylem is supplying it with water and minerals, and actually it's sort of sending sucrose and sugar out of the leaf, I think. That's my guess. Is there anything else we can bring in from the previous tables? Yeah, we could probably mention, you know, the fact that the water content was not different, so the xylem are unaffected, whereas the phloem were affected. So I think that's the conclusion. Okay, let's see what the mark scheme says. Let's see if that was right or not. Okay, so one mark for just kind of describing the obvious. The heat treatment has a greater effect on young leaves than old, possibly because heat treatment damages the phloem. So yeah, okay, and then don't quite follow this point. So Fe3+ moves up. Okay, these 2 points go together, I think. Next 2 points. Fe3+ is transported in the xylem in the older leaf because Fe3+ moves up the leaf into the plant. That's interesting. Yeah, and I think I don't know why these mark scheme points are kind of grouped in a bit of a weird way. I've highlighted the yellow points that I think go together. Heat treatment has a greater effect on the young leaves. Heat treatment damages the phloem, and it suggests that Fe3+ is mostly transported in the phloem in the young leaf. Whereas in the old leaf, Fe3+ moves up the plant in the xylem. Yeah. And then there's also just like general observations that you could have made. I made this observation. There's always, hang on a minute. Yeah, there's always less Fe3+ in the top than the lower parts of the leaves. There's loads of things you could have said here. Loads. Interesting question. It really makes you think. Let's move on. All right, 9.1. Describe the role of two named enzymes in the process of semi-conservative replication of DNA. Okay, so whenever it has like a number of things like that, you've got to go for this like list principle. So if you gave 3 enzymes and the third one was, if the first one was right, they wouldn't mark the third one. So you've got to be sure that your 2 enzymes are good. So what are the 2 enzymes? Obviously, I think you want to talk about DNA polymerase. And probably the other one I would probably say is DNA helicase. You could have also talked about ligase if you'd forgotten about those, but I think those top ones are the best. So DNA polymerase, what does it do? It forms phosphoester or phosphodiester bonds between nucleotides. It doesn't bring the nucleotides together, that's just spontaneously by hydrogen bonding. The DNA helicase unzips the DNA double helix by breaking the hydrogen bonds. Okay, let's look at the mark scheme for the things that you could have said, because I presume there's more than there's a few different enzymes you could talk about, I must assume. Okay, so there's 3 enzymes looks like you can talk about, just 2 actually. Helicase, breaking hydrogen bonds. DNA polymerase joins the nucleotides. You've got to have the word nucleotides forming phosphodiester bonds. This is interesting. Reject hydrolyzes hydrogen bonds. Okay, the reason is a hydrogen bond is not a covalent bond. A hydrogen bond is a interaction between atoms. It's not a covalent bond, so it's not hydrolysis, and there's no water as well involved. Yeah, so interestingly that it doesn't talk about ligase. I guess that's kind of beyond the spec. You might have been taught about it. Ligase joining the backbone when we have the Okazaki fragments. That's kind of beyond the AQA spec. Sometimes they give credit for that sort of thing, but it looks like in this one they didn't. So if you want to bank on some correct answers, talk about helicase and talk about the DNA polymerase. Okay, 9.2. Scientists investigated the function of a eukaryotic cell protein called cyclin A. This protein is thought to be involved with the binding of one of the enzymes required at the start of DNA replication. So this is replication. So this is like initiation of DNA replication. The scientists treated cultures of cells in the following way. Okay, so cyclin A, untreated. Okay, antibody that binds specifically to cyclin A. I'm wondering, is that going to sort of block it or something? Block its activity, maybe? E, added RNA that prevents translation of cyclin A. So so no cyclin A produced, I suppose. No cyclin A protein. And F, added RNA that prevents translation of cyclin A and added cyclin A protein. That's weird. Okay, so there is cyclin A, but it's been artificially added. Huh. They then determined the percentage of cells in each culture in which the DNA was replicating. Okay, well the first thing I'm noticing, okay, control, 91%, that's our baseline. And that is very much the same as F. Okay, so when you kind of blocked cyclin A production but then added more cyclin A, essentially that gets you right back to kind of the control state. What about these? Okay, either of these two messed up DNA replication. Okay, both of these are massively reduced. So if you have the antibody that blocks maybe the binding site of cyclin A, and if you prevent the translation of cyclin A, you have less DNA replication. Okay, so far so good. Seems kind of straightforward so far. What are the questions? Okay, suggest explanations for the results in Table 5. I basically just explained that. So if the protein cyclin A is needed for the initiation of DNA replication. If you block its translation, so there's no protein made in the cell, but you provide an external source of cyclin A, DNA replication can proceed. And there are 2 ways that you can block DNA replication. Number 1, antibody binding, blocking its activity, maybe covering a binding site. I think that's probably what you need to say, because it mentions up here that cyclin A binds to one of the enzymes required. So if an antibody binds over that binding site, then the enzyme required will not be able to bind. Okay, so let's look at the mark scheme, see what you could have said. Oh boy, loads of things. Okay, so okay, so the antibody binds, so it cannot bind to the initiator, etc. That's what we said. Treatment E, RNA I didn't say RNAi. I need to say the RNA interferes with. Okay, RNA interferes with that messenger RNA. In treatment F, the added cyclin A can bind. You need to mention the binding. Yeah, they really want you to say this. You need bind. So much binding is required in this in these answers. I'll pause there. You can have a look through. If you wrote it in a different way, there's so many or different ways you could express this answer. Okay, now we get on to question 10, and this is at the end of every paper. We have these questions that are very knowledge testing. They're very sort of mechanics, kind of content testing. So hopefully this is your opportunity to get 6 marks or 5 marks on these questions for each one, like getting as many as you can. So this one asks you to describe the growth structure of the human gas exchange system and how we breathe in and out. Okay, so first of all, like, analyze the question. So the gross structure of the human gas exchange system. So basically, you want to describe the lungs. It says the gross structure, so that's the big scale structure. So lungs, and maybe go into the alveoli, but we probably want to spend more time on the lungs type stuff. How we breathe in and out. This is like the mechanism of inhalation and expiration. Okay, so maybe kind of try and get 3 or 4. If you tried to get 4 marks for this and 4 marks for this, like a total of 8 marks, then you should get 6, even if you miss, if you don't quite use the right language for one of your points. Okay, so that's what I would go for. I'm just going to go to the mark scheme and run you through it, but I'm also going to put up like a little animation at the same time. Okay, so over here we can see the mark scheme. And over here, I'm going to kind of refer to all these things. So the named structures. Do you need to have all of these? I think you need to have all of these and name them in the correct order for 2 marks. Okay, if you name them and get them in the wrong order, then you get 1 mark. If you put them in the right order, you get 2 marks. So let's go through those structures. So here's the lung. We have the trachea. The trachea is the central kind of windpipe, has rings of C-shaped cartilage that kind of support it and keep it open. The trachea branches into the bronchi. So the trachea has this fork, and then there's 2 bronchi. Off of the bronchi, we get smaller and smaller branches dividing up. Those are called the bronchioles. And the bronchioles, you don't need to know much about the different types of bronchioles, but essentially they get thinner and thinner and thinner, losing that cartilage until ultimately you have these respiratory bronchioles and finally alveolar ducts that lead to the actual alveoli. Okay, so that's kind of points 1 and 2. Now this is mainly focusing on the gross structure, so actually there's not a lot of not many marks for talking about the alveoli and sort of what's going on there at all. In fact, it does say gross structure. That word gross means big, if you didn't know that. Okay, so let's look at this breathing in and breathing out. Right. So, what happens in inhalation and exhalation? So this is the marks 3 and 4 and 5 and in 6. Okay, so there's lots of marks here for this actually. Okay, so inhalation and exhalation. Okay, so let's just click play here. So yeah, this is great. We can see what's going on with all the different parts of the lungs. So first of all, let's just look at the diaphragm. Okay, so in inhalation, the diaphragm contracts and pulls down. So that's this point here. Diaphragm contracts. You need to say and the external intercostal muscles contract. I think this is silly because actually that's not necessarily true. So I don't really like this because actually it depends on whether you're inhaling at rest or if you're inhaling in exercise. Right now I'm sitting down, I'm not doing any exercise, I'm just using my diaphragm. I'm not using my external intercostals to inhale, but I guess that's what you need to say. So diaphragm contracts and the external intercostal muscles contract. The external intercostal muscles, I'll show you them here, are the ones that pull your ribcage up and out. So if you're really inhaling, you pull your ribcage upwards. That's your external intercostals. This causes the volume inside the thorax or inside the ribcage to increase and the pressure therefore decreases. So therefore air moves in from a higher pressure outside into the lungs. When you're breathing out, it's the opposite. The diaphragm relaxes. I'll go back to the diaphragm and kind of springs back up again because it's kind of an elastic tissue. It has elastic fibers in it. And the internal intercostal muscles contract. Again, I really dispute the fact that this is on the MART scheme because I'm not using my internal intercostal muscles to exhale right now. In fact, you only use your internal intercostal muscles to exhale if it's a forced exhalation like this. Normally, even if you use your ribcage to breathe in, it's just gravity that brings it down. So actually your internal intercostal muscles are only really used for forced expiration. So this is actually kind of wrong, but that's the MART scheme. So let's just try and remember that to always talk about both the diaphragm and the intercostal muscles. Okay? Right. Let's move on to the next kind of knowledge-y question. All right, next knowledge-y question. 10.2. Mucus provided by epithelial cells in the human gas exchange system contains triglycerides and phospholipids. Oh, this is so great. This is a lovely question, isn't it? Compare and contrast the structure and properties. Okay, well, this is a nice question, and this. Look at the things that we're asked to do. Compare, contrast, structure, properties. So we might kind of have a little plan almost of, similarities, differences, structure, and properties. Okay, so what are the similarities and what are the differences? Well, let's think about the similarities in structure. Both have glycerol, both have fatty acids, both have ester bonds. What about the differences? Well, the phospholipid has a phosphate group, the triglyceride does not. The phospholipid has 2 fatty acids, the triglyceride has 3 fatty acids. Okay, differences. Properties, triglyceride is generally a hydrophobic molecule all over, whereas the phospholipid, we can call it amphoteric, which means it has a hydrophobic section, that's the fatty acid tails, and a hydrophilic section, that's the head and the glycerol. So the triglyceride molecules do not form bilayers, whereas the phospholipids form bilayers if mixed with water. Okay, so let's look at the MART scheme and check that's everything that we need to say, and I'll illustrate as well. Okay, so let's go through this. So let's look at the 2 structures here. I'm sure you know this already, but let's just go through it. So triglyceride structure, we have the glycerol, and then we do 3 condensation reactions with 3 fatty acids. So there's a fatty acid. And every time we have a fatty acid reacting with that hydroxyl group on the glycerol, we form a ester bond. We can form 3 of those, forming 3 molecules of water with 3 condensation reactions. So then there is the overall structure of the triglyceride, like so, glycerol and the 3 fatty acids. Whereas the phospholipid, if we look down here, it's got the glycerol, it's got 2 fatty acids, 2 ester bonds, and then we have this phosphate group at the top. Now, the properties of this means that it has a hydrophilic section and a hydrophobic section. Okay, and this means that it forms micelles or bilayers when it's in water. Okay, let's look at all the different things you could have said over here. All right, so similarities, ester bonds, glycerol, fatty acids. I don't know if you have to say this bit, maybe saturated or unsaturated, probably should have said. Okay, properties. This is a properties mark. Both are insoluble in water. What atoms there are in them? That's interesting. This is a difference mark. Yeah. Okay. So these are this is the property stuff that I was kind of thinking here. hydrophobic, whereas the phospholipid has hydrophilic and hydrophobic region. And then this is the micelle bilayer thing. Okay, hopefully that was relatively straightforward. Let's move on to the very final question. I think it's the very final question. Yes, it is. Right. Okay. Mucus also contains glycoproteins. So you have to remember what this is. So glycoprotein is a part with a sugar attached to a protein. And they tend to be in the cell membrane. So that's what you need to remember. So it says, describe how lactose is formed, where in the cell it would be attached to a polypeptide to form a glycoprotein. So analyze the question, make sure you do each thing. How is lactose formed? Well, it is a disaccharide, and it is formed from glucose. You could be more specific and say alpha glucose. Alpha glucose? Is it alpha glucose? Is it beta glucose? Can't remember. Glucose and galactose. Hopefully that's worth the mark. I can't remember if you need to specify. We'll see in a second. And where in the cell it would... Okay, so how is it formed? Glucose, galactose, condensation reaction. Okay. How is it formed? Where in the cell it would be attached to a polypeptide to form a glycoprotein. So where? Oh, that's in the Golgi. Golgi apparatus. Probably by another condensation reaction. Worth a go. I'm actually not sure if that's true, but maybe you could say Golgi apparatus, which is where proteins are modified. Okay, right. Let's see what the mark scheme says. Okay, glucose and... Okay, brilliant. Okay, I'm glad I didn't have to remember alpha and beta. I look it up, I've forgotten. Glucose and galactose, it's a disaccharide, joined by condensation. Ah, I forgot to say this, how silly of me. Joined by a glycosidic bond. Say everything you know when it comes to these questions. Added to a polypeptide in the Golgi apparatus. Okay, so I would have got 3 out of 4 for that. Okay, I hope you found that useful. So that was a paper that I did without having really looked at it before. I got most of the marks. At some points I need to work on my phraseology. Perhaps you're in the same boat as me. I'm sort of new to this exam board, so are you. So at this stage, it is about learning the specific things that they often ask you to say and just really like committing those to memory. I will aim to publish another one of these run-through papers in the next week or so, and I'm probably going to do 2019 next. Okay? Good luck revising, and I'll see you next time. Bye-bye.